protein alpha-synuclein begins to misfold and accumulate.
Opening case.
Preclinical alpha-synuclein spread.
The earliest stage of alpha-synuclein-related neurodegeneration rarely presents as a clear neurological disorder. Instead, the first changes often appear subtle and disconnected, affecting functions that are not immediately recognized as part of a unified neurodegenerative process. An individual may notice a slight reduction in the speed of movement. Tasks that require fine motor control, such as buttoning clothing or writing, begin taking longer to complete. The change is gradual and often attributed to fatigue, aging, or reduced physical activity rather than a neurological cause. At the same time, other non-motor symptoms may emerge without clear connection to motor function. A diminished sense of smell is among the earliest and most consistently observed changes. Foods may seem less distinct, and environmental odors may be harder to identify. This alteration occurs without nasal obstruction or infection, making it difficult to attribute to a specific cause. Because olfactory changes do not immediately interfere with daily function, they are often overlooked. Subtle changes in autonomic function may also appear. Gastrointestinal motility can slow, leading to changes in digestion that develop gradually over time. Sleep architecture may become disrupted, including features consistent with REM sleep behavior disturbance, with individuals experiencing disturbances in normal sleep architecture. These symptoms arise in different physiological systems and are rarely recognized as part of a unified process during the early stage. Neurological examination during this phase may reveal minimal abnormalities. Muscle strength remains largely intact, reflexes are often preserved, and coordination appears normal under routine testing conditions. Imaging studies, including magnetic resonance imaging, typically do not show significant structural changes in the brain. The absence of visible pathology reinforces the perception that these symptoms are functional rather than structural. However, a molecular process has already begun within specific neuronal populations. This process does not initially produce widespread neuronal death or produce large-scale anatomical changes. Instead, it affects the behavior of a protein that plays a critical role in synaptic function. The disruption begins at the level of protein structure and spreads through neural networks before significant neuron loss becomes apparent. The protein involved in this process is alpha-synuclein. Under normal conditions, alpha-synuclein is highly expressed in neurons, particularly within presynaptic terminals, particularly at presynaptic terminals where neurotransmitter release occurs. It exists in a soluble form and interacts with synaptic vesicles, contributing to the regulation of neurotransmitter storage and release. This role allows neurons to maintain precise control over synaptic signaling. In healthy neurons, alpha-synuclein remains properly folded and dynamically interacts with cellular membranes and proteins. Its structure allows it to bind to lipid surfaces and participate in the organization of synaptic vesicles. The protein is continuously synthesized, used, and degraded through tightly regulated cellular systems that maintain its concentration within a stable range. The earliest disruption in alpha-synuclein pathology begins when the structure of this protein changes. Instead of maintaining its normal soluble conformation, a portion of alpha-synuclein molecules begins adopting an altered configuration. This misfolded form behaves differently from the normal protein. It loses its ability to interact appropriately with synaptic components and gains the ability to interact with other alpha-synuclein molecules in abnormal ways. At this stage, the number of misfolded molecules remains small. Neurons continue functioning, and synaptic transmission remains largely intact. However, the presence of these altered proteins introduces a new dynamic within the cell. Misfolded alpha-synuclein can act as a template that influences the structure of nearby normal molecules, promoting further misfolding. This process does not remain confined to a single location. Neurons are interconnected through synaptic pathways that allow the transfer of molecular components between cells. Alpha-synuclein, particularly in its misfolded form, can be released from one neuron and taken up by another. This transfer allows the misfolded structure to propagate through neural networks. During the early stage, this propagation occurs at a microscopic level, and does not produce immediate large-scale structural damage. Neurons continue transmitting signals, and the brain maintains functional output through compensatory mechanisms. The subtle motor and non-motor symptoms that appear reflect small inefficiencies within these networks, rather than complete failure. From the outside, the system appears stable. The absence of visible structural degeneration masks the underlying process of protein misfolding and propagation. Yet within synaptic terminals, alpha-synuclein has begun shifting from a functional regulator of neurotransmission to a protein capable of spreading abnormal structure across connected neurons. This early phase represents a critical point in the disease process. The pathology does not begin with widespread neuron death or localized lesions. It begins with the failure to contain a protein within its normal structural state. As misfolded alpha-synuclein begins interacting with other molecules and moving between neurons, the conditions are established for a progressive process that will eventually affect entire neural systems. To understand how this process unfolds, it is necessary to examine the biological architecture of alpha-synuclein and the systems that normally regulate its structure, function, and degradation within healthy neurons. This progression often follows a predictable anatomical pattern described in staging models of alpha-synuclein pathology, biological architecture. Neurons rely on precise control of synaptic transmission to maintain communication across neural networks. This process depends on the regulated release of neurotransmitters from presynaptic terminals, where chemical signals are converted into electrical responses in adjacent neurons. The machinery responsible for this process is highly organized and requires coordination between synaptic vesicles, membrane proteins, and regulatory molecules that control vesicle movement and fusion. Alpha-synuclein is a key component of this presynaptic system. It is a small protein that is highly expressed in neurons, particularly within the terminals of axons where neurotransmitter release occurs. Under normal conditions, alpha-synuclein exists in a soluble, unfolded state in the cytoplasm, but adopts a more structured configuration when it binds to lipid membranes. This ability to transition between conformations allows it to interact dynamically with synaptic vesicles. At the presynaptic terminal, alpha-synuclein associates with the membranes of synaptic vesicles that store neurotransmitters. These vesicles must be precisely organized and mobilized to ensure that neurotransmitters are released in a controlled manner. Alpha-synuclein contributes to this process by regulating the clustering of vesicles and influencing their availability for release. The release of neurotransmitters requires vesicles to move toward the presynaptic membrane, where they undergo fusion and release their contents into the synaptic cleft. This process is mediated by a group of proteins known as SNARE proteins, which facilitate membrane fusion. Alpha-synuclein interacts with components of the SNARE complex and influences the assembly of these proteins, thereby contributing to the efficiency of neurotransmitter release. In addition to its role in vesicle regulation, alpha-synuclein contributes to maintaining synaptic plasticity. Neurons must adapt their synaptic strength in response to activity, which requires changes in vesicle dynamics and neurotransmitter release. By modulating vesicle organization and SNARE complex function, alpha-synuclein participates in the adjustments that allow synapses to strengthen or weaken over time. The concentration of alpha-synuclein within neurons is tightly regulated through a balance between synthesis and degradation. The protein is continuously produced and cleared to maintain a stable intracellular environment. Two primary systems are responsible for degrading alpha-synuclein: the ubiquitin proteasome system and the autophagy lysosomal pathway. The ubiquitin proteasome system targets proteins for degradation by attaching ubiquitin molecules to them. These tagged proteins are then directed to the proteasome, where they are broken down into smaller components. This system is particularly effective for degrading soluble proteins that are properly unfolded and accessible. The autophagy lysosomal pathway handles larger protein complexes and damaged organelles. During autophagy, cellular components are enclosed within membrane-bound vesicles and transported to lysosomes, where they are degraded. This pathway is essential for removing aggregated or misfolded proteins that cannot be processed by the proteasome. Alpha-synuclein also undergoes post-translational modifications that influence its behavior. These modifications include phosphorylation and interactions with lipid membranes. Under normal conditions, these changes remain tightly controlled and do not lead to aggregation. The protein remains soluble and capable of interacting dynamically with synaptic components. The structural properties of alpha-synuclein are central to its function. The protein contains regions that allow it to bind to lipid membranes and regions that can interact with other proteins. Its flexible structure enables it to adapt to different cellular environments, but this flexibility also makes it sensitive to changes in cellular conditions. The maintenance of alpha-synuclein in its functional state depends on cellular systems that regulate protein folding and prevent aggregation. Molecular chaperones assist in maintaining proper protein conformation, while degradation systems remove proteins that become misfolded. These mechanisms ensure that alpha-synuclein remains in a soluble and functional form. Neurons are particularly dependent on these regulatory systems because of their long lifespan and high metabolic activity. Unlike many other cell types, neurons are not easily replaced once damaged. This makes the maintenance of protein homeostasis critical for long-term neuronal survival. The balance between alpha-synuclein synthesis, folding, and degradation represents a key aspect of neuronal stability. When this balance is maintained, alpha-synuclein functions as a regulator of synaptic activity. However, when this balance becomes disrupted, the same structural properties that allow alpha-synuclein to interact with membranes and proteins can lead to abnormal interactions and aggregation. The biological architecture of alpha-synuclein therefore reflects a system that is both highly functional and inherently vulnerable. Its role in synaptic regulation depends on precise control of its structure and localization. When these controls fail, the protein can transition from a soluble regulator of neurotransmission into a misfolded species capable of interacting with other molecules in ways that promote aggregation and propagation across neural networks. Early mechanism. The earliest step in alpha-synuclein-related neurodegeneration begins with a change in the structural conformation of the protein. Under normal conditions, alpha-synuclein exists in a soluble state that allows it to interact transiently with synaptic vesicle membranes. This structure is not fixed, but dynamically adapts depending on its environment. The protein remains functional as long as it can maintain this flexible and reversible conformation. When cellular conditions begin to shift, this balance becomes disrupted. Factors such as oxidative stress, alterations in lipid membrane composition, lysosomal dysfunction, and genetic influences affecting protein handling, and disturbances in protein homeostasis influence the folding behavior of alpha-synuclein. These changes alter the interactions between amino acid residues within the protein, causing it to adopt an alternative conformation that differs from its functional state. This altered conformation exposes regions of the protein that are normally less accessible. These exposed regions have a higher tendency to interact with similar regions on other alpha-synuclein molecules. As a result, misfolded alpha-synuclein molecules begin associating with one another through intermolecular interactions. At the earliest stage, these interactions produce small assemblies composed of a few alpha-synuclein molecules. These assemblies are known as oligomers. Unlike large aggregates that form later in the disease process, oligomers remain soluble and dispersed within the cytoplasm. However, their structural properties differ significantly from those of normal alpha-synuclein. The formation of oligomers introduces a new dynamic within the neuron. Misfolded alpha-synuclein molecules can act as a template-like influence on nearby proteins that influence the folding of nearby normal molecules. When a normal alpha-synuclein protein interacts with an oligomer, the interaction may induce it to adopt the same misfolded conformation. This templated conversion allows the number of misfolded molecules to increase over time. At this stage, the total amount of misfolded alpha-synuclein remains relatively low compared to the total pool of normal protein. Neurons continue functioning, and synaptic transmission is largely preserved. However, subtle disruptions begin emerging at the level of synaptic activity. Alpha-synuclein plays a role in regulating synaptic vesicle organization and neurotransmitter release. When a portion of the protein becomes misfolded, its ability to interact with vesicle membranes changes. Oligomeric forms of alpha-synuclein may bind differently to lipid surfaces, altering the normal clustering and mobility of synaptic vesicles. These changes affect the efficiency of neurotransmitter release. Vesicles may not be positioned optimally for fusion with the presynaptic membrane, and the assembly of SNARE proteins involved in vesicle fusion may become less efficient. As a result, the timing and quantity of neurotransmitter release begin to change. The disruption of synaptic function at this stage is subtle. Neurons remain capable of transmitting signals, but the precision of communication begins declining. Small delays in neurotransmitter release or slight reductions in signal strength can accumulate across neural circuits, producing early functional changes that are difficult to detect through standard examination. At the same time, misfolded alpha-synuclein begins interacting with cellular membranes in ways that can disrupt their integrity. Oligomeric forms of the protein have the ability to insert into lipid bilayers, altering membrane permeability. This interaction can affect ion balance within the neuron by allowing ions such as calcium to move across membranes more freely than normal. Changes in calcium regulation influence multiple cellular processes. Elevated intracellular calcium can activate enzymes that modify proteins and affect cytoskeletal stability. Although these effects remain limited during the early stage, they contribute to increasing cellular stress. Another critical feature of this stage is the potential for misfolded alpha-synuclein to be released from the neuron. Synaptic activity involves the exchange of vesicles and membrane components between neurons. During this process, small amounts of alpha-synuclein can enter the extracellular space. Misfolded forms of the protein can be included in this release. Neighboring neurons can take up extracellular alpha-synuclein through endocytic mechanisms. Once inside a new neuron, misfolded alpha-synuclein can interact with the normal protein pool, promoting further misfolding. This process allows the pathology to extend beyond the original neuron. The propagation of misfolded alpha-synuclein at this stage remains limited and slow. However, it establishes the mechanism through which the disease can spread across neural networks. The process is not driven by cell death, but by the transfer of protein conformation from one neuron to another. The early mechanism of alpha-synuclein pathology therefore involves the transition of the protein from a soluble, functional state to a misfolded form capable of self-association and propagation. This transition introduces both local synaptic dysfunction and the potential for network-level spread. As the number of misfolded molecules increases and their interactions become more stable, additional biochemical changes begin to occur. These changes promote the formation of larger aggregates and further disrupt cellular systems, marking the transition into the next phase of disease progression. Escalation. As misfolded alpha-synuclein accumulates within neurons, the small oligomeric assemblies formed during the early stage begin undergoing structural reorganization. Individual misfolded molecules align along shared beta sheet configurations, allowing them to bind more tightly to one another. This alignment produces elongated structures known as protofibrils, which represent an intermediate stage between soluble oligomers and fully formed fibrillar aggregates. Protofibrils possess greater structural stability than oligomers and are less soluble within the cytoplasm. As additional alpha-synuclein molecules attach to these growing structures, the protofibrils extend in length and begin forming mature fibrils. These fibrils consist of tightly packed protein strands that are resistant to normal cellular degradation processes. The accumulation of fibrillar alpha-synuclein leads to the formation of intracellular inclusions known as Lewy bodies. Lewy bodies are composed primarily of aggregated alpha-synuclein, along with lipids, ubiquitinated proteins, and other cellular components, along with other cellular components, including lipids and ubiquitinated proteins. These inclusions typically form within the cytoplasm of neurons and may occupy significant portions of the cell body. The formation of Lewy bodies represents a transition from dynamic protein interactions to stable insoluble aggregates. Unlike soluble alpha-synuclein, which can participate in synaptic regulation, Aggregated forms become sequestered within these inclusions and no longer contribute to normal cellular function. Instead, they interfere with intracellular organization and place a burden on protein clearance systems. Proteostasis systems become increasingly stressed during this stage. The ubiquitin proteasome system attempts to degrade misfolded alpha-synuclein by tagging it with ubiquitin molecules. However, the tightly packed structure of fibrils prevents efficient processing by the proteasome. As a result, ubiquitinated alpha-synuclein accumulates rather than being degraded. Autophagy pathways also attempt to remove aggregated proteins by enclosing them within membrane-bound vesicles and delivering them to lysosomes for degradation. While autophagy can process some aggregated material, the size and density of Lewy bodies often exceed the capacity of this system. As autophagic efficiency declines, aggregated proteins persist within the neuron. The presence of these aggregates alters the intracellular environment. Lewy bodies displace normal cellular components and disrupt the spatial organization of organelles. Mitochondria, vesicles, and cytoskeletal elements become compressed or misaligned as aggregates expand within the cytoplasm. Mitochondrial function becomes increasingly impaired during this stage. Alpha-synuclein aggregates can associate with mitochondrial membranes, interfering with processes involved in energy production. The electron transport chain, which generates ATP, becomes less efficient under these conditions. Reduced ATP production limits the energy available for maintaining neuronal function. Energy deficits affect multiple cellular systems simultaneously. Ion pumps responsible for regulating membrane potential require continuous ATP to maintain sodium and potassium gradients. As energy supply declines, these pumps operate less effectively, leading to disturbances in ion balance within the neuron. Elevated intracellular calcium levels activate enzymes that degrade structural proteins and contribute to further cellular stress. Synaptic function also becomes more severely disrupted as aggregation progresses. The normal role of alpha-synuclein in regulating synaptic vesicles is lost when the protein becomes sequestered within aggregates. Vesicle organization at the presynaptic terminal becomes disordered, and the efficiency of neurotransmitter release declines further. At the same time, aggregated alpha-synuclein begins interfering with cytoskeletal elements. Microtubules and actin filaments, which support neuronal structure and transport systems, become destabilized as the production of regulatory proteins declines and enzymatic degradation increases. This destabilization affects both axons and dendrites, reducing their structural integrity. The accumulation of aggregates also increases the likelihood of alpha-synuclein being released into the extracellular environment. Neurons under stress may release aggregated or partially aggregated forms of the protein through exocytosis or during cellular damage. These extracellular aggregates can be taken up by neighboring neurons, continuing the propagation process established during the early stage. As propagation accelerates, multiple neurons within a connected network begin accumulating aggregated alpha-synuclein. This spread is not random, but follows established synaptic pathways. Regions with strong connectivity are more likely to become sequentially involved as aggregates move from one neuron to another. The escalation phase therefore represents a shift from localized misfolding to widespread intracellular aggregation and increasing intercellular propagation. The presence of stable fibrillar structures, the formation of Lewy bodies, and the failure of proteostasis systems transform alpha-synuclein from a functional protein into a persistent source of cellular stress. As these aggregates continue expanding and interfering with multiple cellular systems, the neuron's ability to maintain normal function declines further. The next stage of the disease involves the direct impact of these disruptions on synaptic activity, axonal transport, and the structural integrity of neuronal connections. Cellular damage. As alpha-synuclein aggregates accumulate within neurons, the disruption extends directly into the systems responsible for synaptic communication and intracellular transport. The normal function of alpha-synuclein in regulating synaptic vesicles is lost as the protein becomes sequestered within fibrillar aggregates. This loss alters the organization and availability of synaptic vesicles at the presynaptic terminal. Synaptic vesicles must be positioned close to the presynaptic membrane to allow rapid release of neurotransmitters in response to incoming signals. This positioning depends on coordinated interactions between vesicle-associated proteins and the cytoskeleton. When alpha-synuclein is no longer available in its functional form, vesicle clustering becomes disorganized. Vesicles may accumulate away from release sites or fail to mobilize efficiently. The assembly of SNARE proteins, which mediate vesicle fusion with the presynaptic membrane, also becomes less efficient. Alpha-synuclein normally supports the formation of SNARE complexes by interacting with their components. When the protein is trapped in aggregates, this regulatory function is impaired. As a result, vesicle fusion occurs less reliably, and the release of neurotransmitters becomes inconsistent. This disruption affects the strength and timing of synaptic signaling. Signals transmitted between neurons may become weaker or delayed, reducing the efficiency of communication across neural circuits. Although individual synapses may still function, the cumulative effect of these disruptions across thousands of synaptic connections leads to measurable declines in network performance. At the same time, cytoskeletal instability begins affecting axonal transport systems. Microtubules provide the structural tracks along which motor proteins carry vesicles, mitochondria, and protein complexes through the neuron. The integrity of these tracks depends on proper protein synthesis and maintenance of cytoskeletal elements. As protein synthesis becomes disrupted and enzymatic degradation increases, microtubules lose their stability. Segments of the cytoskeleton may become fragmented or misaligned. Motor proteins traveling along these tracks encounter interruptions that slow or block their movement. Vesicles carrying neurotransmitters accumulate within the axon instead of reaching synaptic terminals. Mitochondrial transport is also affected. Mitochondria must be distributed throughout the neuron to supply ATP to regions with high energy demand, particularly synaptic terminals. When transport pathways are disrupted, mitochondria cluster near the cell body, leaving distal regions of the axon and dendrites with insufficient energy supply. Energy deficits have direct consequences for neuronal function. Ion pumps responsible for maintaining membrane potential require continuous ATP. When energy supply declines, these pumps operate less effectively, leading to accumulation of sodium and calcium ions within the neuron. Elevated calcium levels activate enzymes that degrade cytoskeletal proteins, further weakening structural integrity. Alpha-synuclein aggregates themselves contribute to cellular damage by interacting with cellular membranes. These aggregates can disrupt membrane structure, altering permeability and affecting ion balance. Changes in membrane integrity increase cellular stress and contribute to dysregulation of intracellular signaling pathways. Dopaminergic neurons are particularly vulnerable to these disruptions. These neurons, located in regions such as the substantia nigra, rely heavily on efficient vesicle transport and precise regulation of neurotransmitter release. Dopamine synthesis, storage, and release require tightly coordinated processes that are sensitive to disruptions in vesicle dynamics and energy supply. When alpha-synuclein aggregation interferes with these processes, dopaminergic neurons experience a decline in functional capacity. Dopamine release becomes less efficient, and synaptic signaling within motor control circuits begins to weaken. These changes contribute to the early motor symptoms observed in alpha-synuclein-related disorders. The accumulation of aggregates also interferes with protein degradation systems. The ubiquitin proteasome system becomes overwhelmed by the volume of misfolded protein, reducing its ability to degrade other damaged proteins. Autophagic pathways, already strained by the presence of large aggregates, cannot efficiently clear the accumulating material. As these degradation systems fail, additional misfolded proteins begin accumulating within the neuron. This accumulation further disrupts cellular organization and contributes to a broader failure of proteostasis. The neuron becomes increasingly unable to maintain the balance between protein synthesis, folding, and degradation. Dendritic structures also begin showing signs of decline during this stage. Dendritic spines rely on cytoskeletal stability to maintain their shape and support synaptic receptors. As cytoskeletal proteins degrade and energy supply declines, these spines begin shrinking and disappearing. Each lost spine represents the removal of a synaptic connection, reducing the neuron's ability to receive input. Axonal regions develop localized swellings where transport has been disrupted. These swellings contain accumulated vesicles, damaged mitochondria, and fragmented cytoskeletal components. The presence of these swellings indicates areas where the internal architecture of the axon has collapsed. The cumulative effect of these processes is a progressive loss of functional capacity within the neuron. Synaptic communication weakens, transport systems fail, energy production declines, and structural integrity deteriorates. The neuron remains alive during much of this stage, but its ability to sustain normal activity becomes increasingly compromised. Cellular damage in alpha-synuclein pathology therefore arises from the combined effects of synaptic dysfunction, transport failure, mitochondrial impairment, and persistent protein aggregation. These mechanisms interact to destabilize the neuron and prepare the conditions for further amplification of the degenerative process through immune activation and metabolic stress. Immune, metabolic amplification. As alpha-synuclein aggregation and cellular dysfunction progress within neurons, the surrounding neural environment begins responding to signals of damage. Neurons undergoing synaptic failure, cytoskeletal disruption, and metabolic stress release molecular signals into the extracellular space. These signals include fragments of misfolded proteins, damaged membrane components, and intracellular molecules that are normally contained within the cell. Their presence activates the immune surveillance system of the central nervous system. Microglial cells detect these signals through pattern recognition receptors, including toll-like receptors, and transition from a resting surveillance state into an activated phenotype. Activated microglia retract their monitoring processes and begin producing inflammatory mediators intended to manage cellular injury. Cytokines, such as interleukin-1 beta, tumor necrosis factor alpha, and interleukin-6, accumulate within the extracellular environment surrounding affected neurons. Although this immune response initially serves to remove damaged cellular components, prolonged activation of microglia alters neuronal function. Cytokines influence intracellular signaling pathways that regulate protein modification and degradation. In the presence of chronic inflammation, these pathways promote biochemical changes that increase the tendency of alpha-synuclein to aggregate. Inflammatory signaling enhances the activity of enzymes that modify alpha-synuclein, including kinases that alter its phosphorylation state. These modifications reduce the solubility of the protein and promote its transition from soluble forms into aggregated fibrils. As aggregation increases, additional cellular stress signals are generated, further sustaining microglial activation. This interaction establishes a feedback loop between protein aggregation and immune activation. Misfolded alpha-synuclein stimulates inflammation, and inflammation accelerates the aggregation process. Each cycle intensifies the overall level of stress within the neural environment. Astrocytes also contribute to this amplification. Under normal conditions, astrocytes regulate extracellular ion concentrations, recycle neurotransmitters, and provide metabolic support to neurons. When exposed to inflammatory cytokines, astrocytes adopt a reactive phenotype characterized by increased production of inflammatory mediators and altered metabolic function. Reactive astrocytes release chemokines that recruit additional microglial cells, amplifying the inflammatory response. The inflammatory environment leads to increased production of reactive oxygen species. Activated microglia generate these molecules as part of their immune activity. Reactive oxygen species contain unstable oxygen atoms capable of reacting with proteins, lipids, and nucleic acids. In neurons, these reactions produce oxidative damage that affects multiple cellular components. Mitochondria are particularly susceptible to oxidative stress. These organelles generate ATP through oxidative phosphorylation, a process that depends on the integrity of the electron transport chain. Reactive oxygen species disrupt components of this chain, reducing the efficiency of energy production. As mitochondrial function declines, Neurons experience increasing difficulty meeting their energy demands. Energy deficits affect essential cellular processes. Ion pumps responsible for maintaining membrane potential require continuous ATP. When ATP supply decreases, these pumps operate less efficiently, allowing sodium and calcium ions to accumulate within the neuron. Elevated intracellular calcium activates enzymes that degrade cytoskeletal proteins and further destabilize cellular structure. Impaired mitochondrial function also affects the ability of neurons to maintain protein homeostasis. Proteasomal degradation and autophagic pathways both require energy to function effectively. As ATP levels decline, these systems become less capable of removing aggregated alpha-synuclein. The accumulation of protein aggregates therefore accelerates as degradation capacity decreases. Autophagic pathways become increasingly inefficient during this stage. Large alpha-synuclein aggregates are difficult to enclose within autophagic vesicles, and lysosomal degradation becomes impaired under conditions of metabolic stress. As a result, aggregated proteins persist within neurons and continue interfering with cellular processes. The inflammatory environment also influences the spread of alpha-synuclein pathology between neurons. Aggregated forms of the protein can be released into the extracellular space through exocytosis or during cellular damage. Microglial cells may also contribute to this process by releasing partially degraded protein fragments after phagocytosis. Neighboring neurons can take up extracellular alpha-synuclein through endocytic mechanisms. Once internalized, these aggregates interact with soluble alpha-synuclein within the new host cell, promoting further misfolding through templated interactions. This process allows the pathology to propagate along synaptically connected networks. The combined effects of neuroinflammation, oxidative stress, mitochondrial dysfunction, and impaired protein clearance transform alpha-synuclein pathology into a self-sustaining degenerative process. The initial misfolding of the protein has now expanded into a widespread disturbance affecting multiple cellular systems. As this amplification continues, structural damage accumulates within neurons and their connections. The next phase of the disease involves the progressive breakdown of neuronal architecture, where dendrites retract, axons degenerate, and entire neural pathways lose their structural integrity. Structural destruction. As alpha-synuclein aggregation, inflammatory signaling, and metabolic stress continue interacting within neural tissue, the structural architecture of neurons begins undergoing progressive deterioration. The cytoskeleton, which provides mechanical support and maintains the shape of axons and dendrites, becomes increasingly unstable. Microtubules fragment under conditions of impaired protein synthesis and enzymatic degradation, while actin filaments within dendritic spines lose their organization as energy deficits and calcium dysregulation intensify. Dendritic spines are among the first structures to show visible structural decline. These protrusions along dendrites serve as the primary sites of synaptic communication, containing receptors and signaling complexes required for neurotransmission. When cytoskeletal support weakens, dendritic spines begin shrinking and disappearing. The loss of these structures reduces the number of synaptic inputs a neuron can receive, weakening its integration into surrounding neural circuits. As spine density declines, the distribution of synaptic receptors becomes disrupted. Receptor proteins that normally cluster within spine heads become dispersed or degraded, further impairing the ability of the neuron to respond to incoming signals. Each lost spine represents a permanent reduction in connectivity, contributing to the fragmentation of neural networks. Dendritic branches themselves begin retracting as structural instability spreads. Microtubule bundles that support dendritic architecture become fragmented, and sections of the dendrite may thin or collapse. This retraction reduces the total surface area available for synaptic connections, amplifying the loss of communication pathways. Axons undergo parallel structural deterioration. The axon depends on stable microtubule tracks to maintain its elongated structure and to support long-distance transport of vesicles and organelles. When microtubules disassemble, axonal transport becomes severely disrupted. Vesicles carrying neurotransmitters and mitochondria accumulate within the axon, producing localized swellings along its length. These swellings, known as axonal spheroids, indicate regions where the internal architecture of the axon has collapsed. Accumulated vesicles, damaged mitochondria, and fragmented cytoskeletal components fill these areas because transport pathways are blocked. The swelling distorts the axonal membrane and increases mechanical stress, further weakening structural integrity. As degeneration progresses, segments of the axon may fragment. Proteolytic enzymes activated by elevated intracellular calcium degrade proteins responsible for maintaining axonal continuity. Once these proteins are destroyed, the axon separates into disconnected segments, interrupting the pathway required for signal transmission between neurons. The substantia nigra, a region of the brain containing dopaminergic neurons, is particularly affected during this stage. These neurons possess long, highly branched axons that require efficient transport systems and stable cytoskeletal structures. When alpha-synuclein pathology disrupts these systems, dopaminergic neurons experience accelerated structural decline. The loss of dopaminergic neuron integrity affects the basal ganglia, a network of structures involved in regulating movement. Axonal projections from the substantia nigra to the striatum degenerate, reducing the availability of dopamine within this circuit. The structural disconnection of this pathway contributes directly to motor dysfunction. White matter tracts connecting different brain regions also begin deteriorating as axons fragment and disappear. These tracts normally allow rapid communication between distant areas of the brain. As their integrity declines, signals traveling between regions encounter interruptions, reducing the efficiency of neural communication. At the tissue level, these structural changes become visible as reductions in brain volume. Regions with high concentrations of affected neurons show signs of atrophy as dendrites retract and axons are lost. The substantia nigra exhibits decreased neuronal density, and associated pathways within the basal ganglia show reduced connectivity. Microglial cells continue removing debris from degenerating neurons during this stage. While this process clears damaged material, it also eliminates structural remnants that might otherwise support repair. The removal of fragmented axons and dendrites leaves behind areas where the original neural architecture has been permanently lost. Reactive astrocytes contribute to structural remodeling by producing extracellular matrix components that stabilize damaged tissue. However, this stabilization comes at the cost of reduced regenerative potential. The formation of glial scars creates a physical barrier that prevents new axonal growth across affected regions. The cumulative effect of these processes is the progressive dismantling of neuronal architecture. Dendrites retract, synaptic connections disappear, axons fragment, and entire pathways lose their structural integrity. Although some neurons remain present, the network that once connected them has been significantly weakened. This stage marks the transition from cellular dysfunction to large-scale anatomical degeneration. As structural connections are lost, the ability of neural circuits to coordinate activity begins collapsing. The next phase involves the failure of network-level communication as these structural disruptions accumulate across interconnected brain systems. Network-level collapse. As structural destruction progresses through dopaminergic neurons and their connecting pathways, the effects extend beyond individual cells and begin disrupting the coordinated activity of entire neural networks. The brain relies on interconnected circuits that regulate movement, cognition, and autonomic function through synchronized signaling. When alpha-synuclein pathology dismantles the structural connections within these circuits, the timing and reliability of communication begin deteriorating. One of the primary systems affected is the basal ganglia thalamocortical loop, which regulates voluntary movement. This network depends on balanced signaling between the substantia nigra, striatum, globus pallidus, thalamus, and motor cortex. Dopamine released from neurons in the substantia nigra modulates activity within the striatum, allowing precise control of movement initiation and execution. As alpha-synuclein propagation leads to degeneration of dopaminergic neurons, dopamine availability within this network declines. The reduction in dopamine alters the balance between excitatory and inhibitory pathways within the basal ganglia. Circuits that normally facilitate movement become less active, while those that inhibit movement become more dominant. This imbalance disrupts the flow of signals through the network. Motor commands generated in the cortex encounter resistance as they pass through the basal ganglia. The thalamus, which normally relays these signals back to the cortex, receives altered input and transmits less effective output. As a result, the execution of voluntary movement becomes slower and less coordinated. The disruption is not limited to signal strength, but also affects timing. Neural circuits within the basal ganglia rely on synchronized patterns of activity to coordinate movement. As structural connections deteriorate and synaptic transmission weakens, the timing of neuronal firing becomes less precise. Signals that once arrived in coordinated sequences become desynchronized, reducing the efficiency of motor control. White matter degeneration further contributes to this network-level disruption. Axonal pathways connecting the components of the basal ganglia loop become fragmented, introducing delays and interruptions in signal transmission. Even when neurons remain capable of generating electrical impulses, the pathways required to deliver those impulses across the network may no longer be intact. The brain attempts to compensate for these disruptions by increasing activity in remaining pathways. Surviving neurons may strengthen their synaptic connections, and alternative circuits may be recruited to support motor function. However, these compensatory mechanisms place additional metabolic demand on neurons already affected by mitochondrial dysfunction and protein aggregation. As degeneration progresses, the capacity for compensation declines. Neural circuits that once functioned as integrated systems begin fragmenting into smaller, less coordinated groups of neurons. Information processed in one region may fail to influence activity in another due to the loss of connecting pathways. Cognitive networks also become affected as alpha-synuclein pathology spreads beyond motor regions. Cortical areas involved in attention, executive function, and memory rely on synchronized communication across distributed networks. When white matter tracks connecting these regions deteriorate, the coordination required for complex cognitive processing weakens. Neurons within these networks may remain active, but their ability to exchange information becomes limited. Signals must travel through fewer or less efficient pathways, increasing processing time and reducing the precision of communication. This disruption contributes to cognitive slowing and reduced flexibility in thought processes. Autonomic networks that regulate functions such as digestion, heart rate, and sleep also experience disruption. These systems depend on coordinated signaling between central and peripheral structures. As alpha-synuclein pathology affects neurons within these pathways, the regulation of autonomic functions becomes less stable. Another critical aspect of network-level collapse involves the disruption of synchronized neuronal oscillations. Neural networks rely on rhythmic patterns of electrical activity to coordinate communication between regions. These oscillations depend on intact synaptic connections and balanced excitatory and inhibitory signaling. As synaptic loss and structural damage accumulate, the ability of neurons to maintain synchronized oscillations declines. Some regions may exhibit reduced activity due to loss of input, while others may show irregular firing patterns due to disrupted regulation. The resulting desynchronization reduces the efficiency of information transfer across the brain. The collapse of network-level coordination represents a major transition in the disease process. The pathology has moved beyond molecular and cellular disruptions to affect the global organization of neural activity. The brain can no longer rely on its original communication infrastructure to maintain coordinated function. Although some neurons remain alive, their ability to participate in integrated network activity is significantly reduced. The loss of synchronization and connectivity prevents the brain from maintaining the complex patterns of activity required for normal movement, cognition, and autonomic regulation. As alpha-synuclein propagation continues and structural damage accumulates, the system approaches a threshold beyond which recovery becomes unlikely. The next phase involves the point at which neuronal loss, persistent protein aggregation, and network disconnection establish a permanent state of dysfunction within the nervous system. Irreversibility boundary. As alpha-synuclein pathology continues propagating across interconnected neural systems, the brain approaches a biological threshold beyond which recovery becomes no longer feasible. In earlier stages, neurons retain a limited ability to compensate for disruption through synaptic strengthening, network reorganization, and metabolic adaptation. These compensatory mechanisms depend on the presence of intact neurons, functional cytoskeletal systems, and preserved communication pathways between brain regions. The irreversibility boundary is reached when the combined effects of protein aggregation, structural degeneration, and network disconnection exceed the capacity of these adaptive systems. At this stage, several converging processes prevent meaningful restoration of neuronal function. One of the defining features of this boundary is the persistence of alpha-synuclein aggregates within neurons. Lewy bodies and related fibrillar structures remain stable and resistant to degradation. Proteasomal and autophagic pathways, already impaired by metabolic stress and oxidative damage, cannot effectively clear these aggregates. As a result, aggregated alpha-synuclein persists indefinitely within the cytoplasm, continuing to interfere with cellular organization and function. These aggregates prevent the re-establishment of normal protein homeostasis. The balance between protein synthesis, folding, and degradation remains disrupted. Newly synthesized alpha-synuclein may continue entering the aggregation pathway, further expanding the burden of misfolded protein within the neuron. At the same time, the propagation of alpha-synuclein pathology continues across neural networks. Misfolded protein released from affected neurons enters neighboring cells and promotes further misfolding through templated interactions. This Prion-Like mechanism ensures that even regions initially unaffected by the pathology can become involved over time. As propagation advances, multiple brain regions connected through synaptic pathways accumulate aggregated alpha-synuclein. The spread follows anatomical connectivity patterns, progressively involving structures within the basal ganglia, brainstem, limbic system, and cortex. The expansion of pathology across these regions amplifies both structural and functional disruption. Neuronal cell death becomes increasingly prevalent during this stage. Prolonged metabolic stress, calcium dysregulation, and cytoskeletal collapse activate intracellular pathways that lead to apoptosis. During this process, neurons undergo controlled dismantling of their internal components. Cellular debris is cleared by microglial cells, leaving behind regions where neuronal populations have been reduced. The loss of neurons introduces a critical limitation to recovery. The adult central nervous system has minimal capacity to replace lost neurons, particularly within complex networks such as the basal ganglia and cortex. When neurons die, the connections they once formed are permanently eliminated. Axonal pathways are also irreversibly disrupted. Fragmented axons are removed by microglial activity, eliminating the physical conduits required for communication between brain regions. Once these pathways are lost, the brain cannot reconstruct them across long distances. The structural network that supported coordinated activity becomes permanently altered. Glial scar formation further limits the possibility of repair. Reactive astrocytes accumulate in areas of neuronal loss and produce extracellular matrix components that stabilize damaged tissue. However, this process creates a barrier to axonal regrowth. New connections cannot easily form across these scarred regions, preventing the restoration of lost pathways. At the network level, the cumulative loss of neurons and connections disrupts the organization of neural circuits beyond repair. Surviving neurons may attempt to compensate by strengthening existing connections, but these adaptations cannot replace the complexity of the original network. The speed, precision, and integration of neural communication remain permanently reduced. The irreversibility boundary therefore represents the point at which the biological systems required for recovery, protein homeostasis, cytoskeletal integrity, synaptic connectivity, and network coordination, can no longer be restored. The brain transitions from a state of progressive degeneration to one defined by permanent structural deficits. Beyond this threshold, the disease process is no longer driven by the possibility of repair, but by the persistence of dysfunction. The structural and functional changes that have accumulated become the stable condition of the nervous system. The final stage reflects the long-term consequences of this irreversible loss of neuronal integrity and network connectivity, where the brain continues to operate within the constraints imposed by widespread protein aggregation and structural degeneration. Stable aftermath. After the irreversibility boundary has been crossed, the nervous system enters a long-term state defined by permanent structural and functional deficits. The propagation of alpha-synuclein may continue at a reduced rate, but the architecture of the brain has already been fundamentally altered. Neurons have been lost, axonal pathways have fragmented, and the regulatory systems that once maintained protein balance remain impaired. Surviving neurons continue operating within this altered environment. Many contain intracellular inclusions composed of aggregated alpha-synuclein. These inclusions persist within the cytoplasm and interfere with intracellular organization. Although some neurons remain viable despite the presence of aggregates, their ability to maintain efficient transport, energy distribution, and synaptic function is reduced. Axonal transport remains compromised in surviving neurons. The microtubule network, which depends on stable protein synthesis and cytoskeletal integrity, cannot fully recover in the presence of persistent aggregation and metabolic stress. Vesicles, mitochondria, and other cellular components move through the neuron with reduced efficiency, limiting the cell's ability to support synaptic activity. Mitochondrial distribution continues to be uneven. Regions of the neuron with high energy demand, particularly synaptic terminals, may receive insufficient ATP. This energy deficit reduces the reliability of neurotransmitter release and weakens the neuron's ability to maintain membrane potential. Electrical signaling may still occur, but with reduced consistency and efficiency. Synaptic density across affected brain regions remains significantly reduced. Dendritic spines lost during earlier stages of degeneration do not regenerate at levels sufficient to restore original connectivity. Each lost spine represents a permanent reduction in communication capacity between neurons. Surviving circuits must operate with fewer synaptic connections, limiting the complexity of neural processing. The basal ganglia network continues to function in an altered state. The loss of dopaminergic neurons in the substantia nigra reduces dopamine availability within the striatum. This deficiency disrupts the balance between excitatory and inhibitory pathways that regulate movement. Motor signals originating in the cortex encounter persistent resistance within this network, resulting in reduced efficiency of movement control. White matter pathways connecting different brain regions often remain partially degraded. Axonal fibers that were lost during earlier stages of degeneration cannot regenerate across long distances. The absence of these pathways limits communication between cortical and subcortical regions. Neural circuits that once operated as integrated systems now function with reduced coordination. Cognitive and autonomic systems may also remain affected, depending on the extent of alpha-synuclein propagation. Cortical regions involved in executive function, attention, and memory may retain some activity, but their ability to communicate with other regions is diminished. Autonomic pathways regulating functions such as digestion and sleep may continue operating with reduced stability. The immune environment within affected regions remains altered. Microglial cells often remain partially activated, continuing to monitor and clear residual cellular debris. Low levels of inflammatory signaling persist, maintaining a biochemical environment that differs from that of healthy neural tissue. This persistent activation contributes to ongoing cellular stress within surviving neurons. Neural plasticity allows some degree of adaptation. Remaining synaptic connections may strengthen, and alternative pathways may partially compensate for lost functions. However, this adaptive capacity is limited by the availability of intact neurons and structural pathways. Compensation cannot fully restore the original architecture of the network. The long-term outcome of alpha-synuclein propagation reflects the persistence of structural loss and molecular dysfunction. What began as a localized misfolding of a synaptic protein has progressed into a widespread disruption of protein homeostasis, cellular integrity, and network coordination. Alpha-Synuclein, which normally functions as a regulator of synaptic vesicle dynamics, becomes a source of pathology when its structure is altered and allowed to propagate between neurons. The failure to contain this protein within its functional state leads to aggregation, spread, and progressive degeneration across neural systems. The stable aftermath represents a condition in which the brain continues functioning with reduced connectivity, diminished metabolic resilience, and persistent protein aggregates embedded within neuronal cytoplasm. Neural activity persists, but the efficiency and coordination of that activity are permanently altered. The system that remains is not inactive, but it operates within the constraints imposed by structural degeneration and ongoing molecular imbalance. The original network architecture that supported precise and coordinated function cannot be restored. This is pathology of fear, and this fear is real.
Pathology of Fear is a medical documentary series presenting clinically documented neurologic and pathological mechanisms for educational analysis.